So we divided the anti-cancer drugs into two major parts. One was cytotoxic, which we covered in the previous video. And in this video, we will be covering the targeted anti-cancer drugs. So basically, we will find a target inside the cancer cells and we will make a drug against those targets. Okay. So we can broadly divide them into two categories. One are called as small molecule inhibitors, and second are called as monoclonal antibodies. Now, as the name tells, these are large in size because they are antibodies, and large drugs cannot cross the membrane. So these are all injectable drugs. These are all injectable. Whereas small molecule inhibitors, they may be given orally depending upon their own properties, like whether they are absorbed or not. Okay. So we'll discuss under these two headings, starting with these small molecule inhibitors. Small molecules we will start. The most important group of drugs in this category are called as tyrosine kinase inhibitors. Tyrosine kinase inhibitors. So from where we reach these drugs, we'll try to understand that there is a chromosome which is known as chromosome 9. We have another chromosome in the body which is called as chromosome 22. Now, one chromosome contains a gene called as ABL. The second chromosome contains a gene called as BCR. And we know that normally these are present on a different chromosomes, and we know chromosomes or the DNA do not talk to each other. They keep on doing their own function. So this chromosome or ABL gene keep on working, this BCR gene keep on working separately. There is no interaction. The person is normal, absolutely normal. Okay? Now once what happened, there was translocation. So chromosome 9 and 22 break and come to lie on same place. So that is translocation 922. Translocation 922 that occurred, and this lead to fusion of these two genes. So ABL and BCR, they come together. This is known as Philadelphia chromosome. Philadelphia chromosome, you all know, in which there is 922 translocation, ABL and BCR fuse together. And when ABL and BCR fuse together, that lead to activation of an enzyme known as tyrosine kinase. And when tyrosine kinase is activated, it lead to uncontrolled growth of cells that resulted in chronic myeloid leukemia. So in the pathogenesis of CML, it was found that there was ABL BCR fusion that activated a tyrosine kinase that lead to leukemia. So we thought that if tyrosine kinase was causing the leukemia, if we inhibit this tyrosine kinase, can we treat CML? And we started developing the drug on this line, and the first drug we developed was imatinib. So we developed a drug called as imatinib. It inhibited this tyrosine kinase, and it was found to be highly effective in CML. It is so much effective that now it is a drug of choice for CML. And then we did the further research, and it was found that this tyrosine kinase is overactive not only in CML, but it is overactive in most of the human cancers. The pathway which is activating a tyrosine kinase may be different. So different pathway which activated tyrosine kinase cause a different cancer. So if we inhibit the tyrosine kinase activated by different pathways, we can treat different cancers. So this led to development of a group of drugs which are called tyrosine kinase inhibitors. So now we have more than 50 drugs in this group. So what are the general points about tyrosine kinase inhibitors? The name of these drugs will end with nib, like imatinib. The name of all will end with nib. Very important to remember, all of these are orally effective drugs. They are oral drugs. Number 3, they are metabolized by CYP3A4. So these are prone to drug interactions. These are prone to drug interactions. Inducers may increase their metabolism, inhibitors may decrease their metabolism, and all these drugs can cause skin rash as a side effect. So that is common to all tyrosine kinase inhibitors. Okay? So the important ones you need to remember. So we can quickly discuss tyrosine kinase inhibitors which are used in chronic myeloid leukemia. So you can remember that CML is common in India. So I and D, the drugs are imatinib, nilotinib, and dasatinib. They are used in chronic myeloid leukemia, and we already discussed imatinib is a drug of choice for CML. Then tyrosine kinase inhibitors, which are used in lung carcinoma, lung cancer. So you can remember that lung cancer, very close to lung is our heart. So lung cancer is treated after ECG. Lung cancer is treated after ECG. What do we mean by this is AF stand for afatinib. Afatinib. E is erlotinib. C is seritinib. And G is geftinib. So these are tyrosine kinase inhibitors which are used in lung cancer. So you can remember lung cancer is treated after ECG. Then we have tyrosine kinase inhibitors for renal cell carcinoma. So patient of RCC, we say pass. You are pass or you go. RCC patient, we pass. Pass stand for pazopanib, axitinib, sunitinib, and sorafenib. So RCC, renal cell carcinoma patient, we pass. But if a patient has hepatocellular carcinoma, we say sorry, we cannot do anything. For HCC, we say sorry. Sorry means sorafenib. Sorafenib. So sorafenib, it is used for hepatocellular carcinoma, and it is in fact now the drug of choice for hepatocellular carcinoma. Sorafenib. Then we have tyrosine kinase inhibitors for GIST, gastrointestinal stromal tumors, and you can remember them as SIR. Remember, whenever in Hindi we say G, we say G sir. So G sir means GIST, we have sir. S for sunitinib, sunitinib. I is imatinib, and R is regorafenib. They are used for GIST, and important to remember, imatinib is a drug of choice for GIST. So imatinib is drug of choice for CML as well as GIST. Then we have tyrosine kinase inhibitor for colorectal cancer. So colorectal, so you can remember colorectal contains R for rectal, R for regorafenib. Regorafenib is used in colorectal cancer or simply rectal carcinoma. Then we have tyrosine kinase inhibitors for malignant melanoma. Malignant melanoma. So we remember these as deep vein thrombosis. So patient of malignant melanoma can develop DVT. So the drugs are dabrafenib, vemurafenib, and tremelimumab. So dabrafenib, vemurafenib, and tremelimumab, they can be used in malignant melanoma. Then we have tyrosine kinase inhibitor for breast cancer. Breast cancer. The drugs are lapatinib and niratinib. Lapatinib and niratinib, they are used for breast cancer. You can remember there is a female called Neera. Neera has lump in the breast. So Neera has lump in the breast. So niratinib and lapatinib, they are used in breast cancer. Okay. Then lastly, we have tyrosine kinase inhibitor for medullary carcinoma of thyroid, and the name of the drug is vandetanib. For medullary carcinoma of thyroid, we have vandetanib, and you can remember when we say Vande, the other person will say Matram. So Vande Matram. So vandetanib, we have medullary carcinoma of thyroid. Okay, so these are the important tyrosine kinase inhibitors. I know you are feeling how you will be able to remember. Yes, it is difficult, but if you see carefully that most of the tyrosine kinase inhibitors, the names are on different letters. The name is ending with nib, and from where it is starting, that will help us in answering the MCQ even without remembering the name. So you just remember the mnemonic. Most of the time you will come to the answer, like CML, common in India, imatinib, nilotinib, dasatinib. Lung cancer, after ECG, after is AF. Remember, this is AF means afatinib, erlotinib, ceritinib, geftinib. On the other hand, RCC pass, the A is different, not AF, F is not there, so it is exitinib. So we have pezopia, suniti, and sorafenib. HCC, sorry is easy, sorafenib. Gist is GSER, sunitinib, imatinib, regorafenib. Colorectal, R4 regorafenib, that is easy. Malignant melanoma, DVT. Breast cancer, Nira has lump in the breast, neratinib and lapatinib, and Vande Matram, vendetanib is for medullary carcinoma, thyroid. Yes, I think you will be able to remember. You just revise 2 or 3 times, you will be able to remember. Okay, so first group of small molecule inhibitors is tyrosine kinase inhibitors. Okay, then we have a second group which is known as proteasome inhibitors. Proteasome inhibitors. Proteasome is an enzyme which is required for cell growth. When we inhibit that, the cell cannot grow, so cell dies, we can treat the cancer. The drugs which are proteasome inhibitor, they are used in multiple myeloma. They are used in treatment of multiple myeloma. The drug examples are bortezomib. The name of the drug will end with zomib. Bortezomib, carfilzomib, and ixazomib. And you can easily remember if you see the name Zomib. Zomib, if you put the I at the last, it will become zombie. So these are zombie drugs. And if you have seen the English movies with the zombies, all the zombies have these hand, foot broken. So hand, foot are broken. So that means present when there is hand, foot are broken in fractures like in multiple myeloma. So zombie drugs are used for multiple myeloma. Okay. Then we have third group of small molecule inhibitors. These are called as PARP inhibitor. PARP inhibitor. PARP is poly ADP ribose polymerase. So this is the another enzyme which is required for growth of the cell. So when we inhibit this, cell cannot grow, the cell will die. That will lead to treatment of cancer. And the drug we use for this purpose is olaparib. Olaparib. Olaparib, the name says that it is a poly ADP ribose polymerase inhibitor. So parib, the name says it is poly ADP ribose polymerase inhibitor, and it is used for O means ovarian cancer. Used for ovarian cancer, olaparib. Okay, so PARP inhibitor are the group. Then the next important group is cyclin dependent kinase 4 and cyclin dependent kinase 6 inhibitors. So these are the important kinases which are important to regulate the cell cycle. So when we inhibit this, we can treat the cancers. The example of the drug is palbociclib. Palbociclib. So from the name you can remember cycli. Cycli means cyclin. Cyclin dependent kinase inhibitors. So cyclibs are cyclin dependent kinase inhibitors, and they are ending with B means they are used for breast cancer. They are used for breast cancer. They are effective orally. So they are oral drug for breast cancer, palbociclib. Another new drug which has formed in this category is ribociclib. So cyclibs are cyclin dependent kinase inhibitors. They are used in breast cancer. They are oral drugs. Okay. So these are the important points regarding the small molecule inhibitors. Okay. Now the second group of drugs are monoclonal antibodies. Now monoclonal antibodies, all of them are injectable. We cannot give them orally. All of them are injectable. So the monoclonal antibodies we will discuss in detail in a separate video. Here the important ones you need to remember. So monoclonal antibody may be against epidermal growth factor receptor. We have monoclonal antibody against epidermal growth factor receptor, and these drugs are cetuximab and panitumumab. Cetuximab and panitumumab, they are against the epidermal growth factor receptor. So these drugs, they are used for C means colorectal carcinoma. They are used for colorectal carcinoma, and you can remember that C means colorectal carcinoma, and this drug contains pani in the name. Pani is water. So when we drink water, it will also go to colon. So it will treat colorectal carcinoma. These are monoclonal antibody against epidermal growth factor receptor. Then we have monoclonal antibody against HER2. You all know HER2 is overactive in which cancer? Yes, breast cancer. So the drugs which are used in breast cancer are trastuzumab and pertuzumab. Trastuzumab and pertuzumab, they are monoclonal antibody against HER2, and these are used in breast cancer. And you can remember from the name again, trastuzumab. If instead of T I write B, it become brastuzumab. So you can remember it is for breast cancer. Then the name is starting with trust. So trastuzumab name start with the trust. So when we trust someone, our heart will be broken. So that will tell us that the major side effect of these drugs is cardiotoxicity. So trastuzumab and pertuzumab are cardiotoxic. So they are against HER2. Then we have monoclonal antibody against platelet derived growth factor receptor. The drug is olaratumumab. Olaratumumab. This is a drug used for soft tissue sarcoma. Soft tissue sarcoma. Olaratumumab is used for soft tissue sarcoma. Then we have monoclonal antibody against CD20. Monoclonal antibody against CD20. The name of the drug is rituximab. Rituximab. It's a monoclonal antibody against CD20, and this is a drug which is used in non-Hodgkin's lymphoma and chronic lymphoid leukemia. Non-Hodgkin's lymphoma and chronic lymphoid leukemia. Okay, so these are the important drugs. Now, once I had a physical live class at Delhi. So I had scheduled the class, but there was a girl called Ritu. So that girl was from a VMMC college, Safdarjung Hospital. So she called me and said that, sir, I had my internship going on. So if I go to the duties, I will come late to your class. So what is the solution? So I said in Hindi, Ritu, you do one thing. What you do is take a CL. अगर तुम CL ले लोगी तो नहीं होगी लेट. So you need to understand little bit Hindi regarding this story. Now, why I am telling you this story? कि if Ritu means rituximab. अगर रितु CL लेगी तो नहीं होगी लेट. So rituximab is used for CLL and non-Hodgkin's lymphoma. Okay. And further, you can remember that if she is coming to my class, she obviously will be of second, third, fourth year, near about 20 years of age. So it is against CD20. Yeah, so CD20 rituximab. Okay. So these are some of the important monoclonal antibodies. Okay. Then moving to one last concept in targeted drugs, these are called immune checkpoint inhibitors. Immune checkpoint inhibitors. So first we'll try to understand what is the meaning of this. Now this is our T cell. We know the function of T cell is to kill the tumor cell. It kills many other cells also, but tumor cells also. So this is a tumor cell. So T cell will kill the tumor cell. But the problem is T cell is killing a tumor cell, but not a normal cell. So what is the difference between a tumor cell and a normal cell? We know mostly the tumor cells are very similar to normal cells. So how it happens that T cell will attack only the tumor cell, not the normal cell? So what is the difference is, this is a tumor cell. This is a tumor cell. What is the difference is, a normal cell, it has been given an ID card. So whenever T cell try to kill a normal cell, the normal cell will show the ID that I am part of your body. Please don't kill me. So T cell will stop. But tumor cells do not have that ID. So what is that ID? Remember, on the T cell there is presence of a receptor called as programmed death receptor, so called as PD1, programmed death receptor. Okay? Programmed death receptor is present on the T cells. So whenever T cell try to attack a normal cell, the normal cell has programmed death ligand 1. Ligand 1. So this ligand will bind to the PD1 of the T cell. So when this bind, the T cell thinks, or T cells finds that this cell is a normal cell, I should not kill this cell. So basically whenever this interaction occurs, the T cells are inhibited. So the immunity is kept under check. Immunity is kept under check against the normal cells because this ID card is given to the normal cell. The name of the ID card is PDL1. So tumor cells do not have this ID card. So whenever T cell attack a normal cell will show the ID card, so T cell will stop. Immunity will stop. On the other hand, when T cell try to kill a cancer cell do not have ID, so it can kill the cancer cell. Okay, so that is the normal thing, normal physiology, normal immunity. But now the cancer cells have become smart. So they came to know that the normal cells survive because they have ID card. So what they do, they make a fake ID card. So now the tumor cell has also started expressing the PDL1. So the tumor cell is having PDL1, then the PD1 which is present on the T cell, it will bind to this also. So again the T cell will be inhibited. So now the T cell is not able to kill the tumor cell, that's why tumor is growing. Yeah. So the tumor cells or cancer cells, they have made a fake ID. So if we stop this interaction, this ID interaction, then we can kill the cancer cell. So that mechanism is known as immune checkpoint inhibitors. So this is the immune checkpoint against normal cells. So what we have done, we have developed two type of drugs, one which can bind to PD1. So if a drug bind to PD1, it will not be able to recognize the ID card. Second, we can give a drug which bind to PDL1. Again, now the ID card cannot be shown to the T cell. So two type of drugs. If we stop this interaction, T cell cannot be inhibited. When T cell is not inhibited, they will kill the cancer cells. That is the concept. Is it okay? So now what are the drugs? The drugs are remembered as new drugs act at immune checkpoint. New drugs act at immune checkpoint. These are the drugs. So N for nivolumab, nivolumab. D for durvalumab, durvalumab. A for avelumab, avelumab. AT for atezolizumab, atezolizumab. I for ipilimumab. C for semiplimab. And P for pembrolizumab. So these are the immune checkpoint inhibitors. So all the immune checkpoint inhibitors I have covered. Question is being asked on immune checkpoint inhibitors. Now discussing the uses of these drugs, although I don't want you to remember the uses, the gross uses or the special uses you can remember, although nivolumab can be used for many types of cancers like lung carcinoma and many others, but the special thing you need to remember, it is used for Hodgkin's lymphoma. Hodgkin's lymphoma. Then durvalumab, it is used for bladder cancer, urinary bladder carcinoma. From the name you can remember, it is saying durva. So durva, if we write bla in front, it becomes bladderva. It is used for bladderva cancer. So bladder cancer. Avelumab is used for Merkel cell carcinoma. Merkel cell carcinoma. So if you remember, in the South African team, there was a cricket player called as LB Morkel. Remember, have you heard the name LB Morkel? LB Morkel was present. So LB means remove the L, it become Abe. So avelumab is used for Merkel, means Merkel cell carcinoma. Avelumab for Merkel cell carcinoma. Then we have atezolizumab. This is also for urothelial carcinoma or urinary bladder carcinoma. Urothelial carcinoma, we have atezolizumab. Ipilimumab is for malignant melanoma. Malignant melanoma, we have ipilimumab. Semiplimab is for squamous cell carcinoma of skin. So you can remember C for cutaneous cancer, skin cancer, and pembrolizumab. This is a drug which is having maximum wide spectrum, can be used for almost every cancer. So you can see pembrolizumab can be used for renal cell carcinoma, hepatocellular carcinoma, breast cancer, malignant melanoma, B-cell lymphomas, lot of cancers. The special thing to remember, it is also used for endometrial carcinoma. No other drug in this group is effective in endometrial carcinoma. Pembrolizumab is used for endometrial carcinoma also. It is used for breast cancer also. So in the name you can find many, but I don't want you to remember. We have endometrial carcinoma, breast carcinoma, lymphoma, we have malignant melanoma, we have breast cancer. Many cancers we can use, but important to remember, pembrolizumab has very wide spectrum. Okay, so these are the important points regarding the immune checkpoint inhibitors, or we can say we have covered the targeted anti-cancer drugs.